Hi, and welcome to this video where we're going to be running through all the things that you need to know for OCR A-Level Biology Module 3.1. So that's the module on exchange surfaces and also breathing. So here's the kind of overview of the Mindmap, what we're going to look at. We're going to look at some stuff to do with surface area and why we need to have exchange systems. We're going to look at the main human exchange system, which is the lungs, or the structure of the lungs and some of the different tissues that you'd find in there, including microscope images of those. Then we're going to be looking at, also to do with the lungs, how we can track breathing using a respirometer and some of the graphs that we'll get with those. We're also going to look at exchange systems in fish and in insects and how those systems work differently. So I've set you this on Teams, these images. There's the A3 version, and then here behind me now is the A4 version, which you can cut out the images and use them to make your mind map. Okay, so if you need to pause the video now, get your resources, get colourful pens, pencils, get your textbook, do so now, and then we're going to get started. Okay, so the first thing we're going to look at over here is why we actually need exchange surfaces. Okay, so at a very basic level, living things need to exchange things with their surrounding environment because we need to get certain molecules and take them into the body and get rid of certain other molecules. So that's really a kind of a why. So we can kind of look at that. So we need to take in oxygen, for example, and get rid of waste products. We also need to take in glucose as well. But really, in this module, we're mainly focused on oxygen. In other modules, well, we don't really look at the digestive system in any great detail at A-level, but a lot of the things that we talk about in terms of exchange surfaces that are true for the lungs will also be true for exchange surfaces in the intestine. And when we're talking about waste products, in this module, we're going to be talking about getting rid of carbon dioxide, but in year 13, you'll also talk about the exchange surfaces that we use to get rid of other waste products, such as the kidneys, which are an excretory surface, but they're also sort of they're to do with getting rid of waste products. Okay, so the major factors that affect the need for exchange surfaces are three things. Well, up here, we're going to talk about surface area to volume ratio, which is really, really important. Okay, so this little box up here, we're going to be talking about that, surface area to volume ratio. And a sort of interrelated thing is size. And we're also going to be talking about metabolic activity, level of activity, let's call it, level of activity. Okay, so let's look at the surface area to volume ratio first of all, okay? So this table here is taken in, it's from your textbook, it's page 170, but we can calculate things here. Surface area in millimeters squared. If you use this little formula here, 4 pi r squared. Now, if the radius in millimeters is 1, can you calculate the surface area of a sphere with the radius 1? Okay, and then can you calculate the volume, and can you calculate the surface area to volume ratio? Okay, use these formulas here. Try and do them, first of all, without me telling you them, because you need to be able to do these things. And then when you get the ratio, I'll give you the answer for the first one, see if you get it right. This one should be 3 to 1, okay? It should always be, these ratios should always be to 1. So the end bit, the volume, always is to 1, okay? So that's something you should bear in mind. See if you can work out these ratios, fill out this table, pause the video, and then I'll give you the answers. Okay, how have you done those? I'm just going to take the answers straight from here. So this is sort of slightly, I'm pinching a bit. Okay, so here we go. The answers taken from your book are, I'm going to round them slightly. I'm not going to give them to quite so many decimal places, but 12.6. And here is 4.2 if I round that up. This one is 50.3 to 1 decimal place. And down here we've got 523.7. Okay, so the key thing is as we get larger, what happens to the surface area to volume ratio? Well, start is 3 to 1, and then by here we have 1.5 to 1, and by here we have 0.6 to 1. Okay, so that is the key conclusion which we should write here. Change size there. So as the object, any object, gets larger, the SA to V, so that's the surface area to volume ratio, decreases. Okay, that is a really, really fundamental and important point. So this means that small objects have a large surface area to volume ratio, and large objects tend to have a a much smaller surface area to volume ratio. So that means that small objects can sometimes get by without a specialized exchange surface because they've got enough surface area. So this kind of plays in here. So this is also linked to size. Okay, so this is how size and surface area volume ratio kind of work together. So small objects have a big surface area to volume ratio. So what does that mean? Well, things like an amoeba, which is smaller than a millimeter. It's a large cell. I think it can be just about up to a millimeter, but they don't need a specialized exchange surface. So things around about a millimeter can get by without having a specialized exchange surface. So I'm going to write x equals less than 1 millimeter. Don't need ES for exchange surface, okay? They don't need it. They can get by. Whereas big things do. Okay, so I'm going to put that over here. So big things, bigger than this, bigger things need exchange surfaces. And there's a little but here. Okay, sometimes shape can affect it. Okay, so shape. This is kind of to do with surface area to volume ratio, actually, because things which have a flat shape have a larger surface area to volume ratio than if they are perfectly spherical. So this is a sort of slightly, I'm going to try and draw a very bad sort of cartoony drawing here of a flatworm. Okay, flatworms don't have eyes like this, but I'm just drawing it so that it's kind of cartoony. This is a flatworm. Flatworms could be 1 or 2 millimeters thick, but maybe about this long or so. And they might not need a specialized exchange surface because they've got that large surface area of their skin, which they can, because they mainly live in the water, they can take in oxygen, let's say, through their skin. But shape, flatworms may not need ES, exchange surface. Okay, there's a flatworm here. The other thing is level activity. Low levels of activity may mean that you don't need an exchange surface because you don't need to take in as much oxygen and don't need to get rid of as much carbon dioxide. But high levels of activity equals you need an exchange surface. Okay, one more thing I think we should put here, and let's take it this way. Yeah, I'm going to take it this way. What is a good exchange surface? Any exchange surface, be it the small intestine, the lungs, the gills, and all sorts of other exchange systems like the gas exchange system in insects, they need to have certain features, okay? So here they are. Speaking of insects, fly in front of me. Right, okay, so good exchange surfaces. What do we need? Well, we need a large surface area. We need short diffusion distance. And we need a good blood supply. So large surface area gives more space across which diffusion can occur. Short diffusion distance means diffusion can happen quickly from one side to the other. And a good blood supply, this maintains a high concentration gradient, okay? Because remember, those are the things that affect diffusion. So maybe add a little note there, just that it kind of maintains the gradient. And I don't want to go too far this way because I'm going to start to encroach on the fish, which we're going to talk about later. Right, now one good exchange surface that we're going to look at next is the lungs, okay? So now we're going to move down to this lower part of the mind map down here. Okay, before we get into the lungs, I thought actually we should write down just a few examples of these good exchange surfaces or exchange surfaces, just so you're aware. Okay, we've got the intestines. We've got the microvilli in the small intestine, which have high surface area, short diffusion distance, and good blood supply. We've got the lungs, of course, which we're going to look at in detail. We've also got the gills, which are going to be over here, and the insect trachea system. What else have we got? We've also in plants, okay, plants, let's do green for plants. So we've got the leaf. The leaf is an exchange surface because that's where carbon dioxide is absorbed for photosynthesis and oxygen is released, vice versa for respiration. We've got the stomata, we've got the spongy mesophyll, that's an exchange surface. And we've also got the root hair cells, or the root system, which again has high surface area, short diffusion distance, things like that. So yeah, just be aware that there are multiple exchange surfaces that follow this pattern of large surface area, short diffusion distance, and good blood supply. Okay, now let's get down into the lungs down here. Okay, so first of all, let us label the lungs. Okay, so here we've got the voice box, which isn't really part of the lungs, that's just what enables us to speak. You can feel it if you put your sort of hand here. It's there, that's the voice box, and below that is the trachea, which you can just, don't do this too hard because it will kind of feel weird. Oh, it feels weird. But if you do that, oh, you can kind of feel the cartilage, but be careful because it feels a bit, you can feel the cartilage on the trachea. So this is the trachea, and here's a little zoomed in version of it. So the trachea contains rings of cartilage. To support it. And that means that when you inhale sharply like that, if you ever try to suck sharply down a straw, sometimes the straw collapses and then the two sides of the straw, instead of being circular, they stick together, and then the water, if you're drinking water, won't flow up the straw. That would be catastrophic. If you were to breathe in sharply and your trachea was to close and shut, that would be catastrophic. So these rings of cartilage keep the trachea open. And I don't know what that arrow is supposed to be pointing to there. I think maybe it's maybe the tissue or the inside, the endothelial tissue and the inside the trachea there. But the rings of cartilage, also we can, they're also C-shaped rings of cartilage. Now, I'm not really sure. I think, okay, so when you swallow food, I think that the esophagus is behind the trachea, not in front, so it's behind it. So when you swallow like a really big bit of food, like a big lump, you haven't chewed your food properly, as that, it's called a bolus, lump of food, as it's going down your esophagus into your stomach, it kind of presses against the trachea. So the C shapes, if this is a C, the C shapes kind of face this way of cartilage in your trachea, so that the food can move down behind it without kind of, there's a bit of flexibility in the bit with the C, where there's a gap. Okay, it's an interesting fact about swallowing food. Okay, so trachea, voice box, let's label the rest down here. I'm just going to zoom in a tad. Okay, now difficult to look. Okay, so as the trachea goes down, 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 it then splits. So I'm actually going to also label the flow of air as well. So air goes down, 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 and then it splits. Okay, it can split this way and that way. Okay, that didn't work quite as I wanted to. Let's do it this way. Okay, down, 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 and then it splits like this. When it splits, it goes not into the trachea, but into the bronchus, okay? There's two, okay? The left bronchus and the right bronchus. Left, right. So this actually here, this side, that would be, if you're looking at the person, that would be their left bronchus, and the right would be on this side of the diagram over here. Okay, so bronchus there. Here we've got a rib. So the rib cage, let's call it rib cage. This is one rib there. But the rib cage surrounds this whole thing. And can we actually see, so these bits in here, this darker bit there, I'll label it there, that is the intercostal muscles. Oops. Intercostal muscles. Those muscles lie between the ribs and they are involved in breathing. More on that in a minute. Okay, so once we go past the bronchus, we get to the bronchioles. Now, I'm actually going to label them over here. And it's not the final gap there, but it's this kind of this thing there. Okay, can you see that? It's this tube. So we go from bronchus into smaller tubes called bronchioles, and at the very end we get to the alveolus, which is this little circular bit here. So I'm going to label there. So that's the alveolus. Alveolus is the singular. Alveoli is the plural. So there is a zoomed in version of alveolus or alveoli, and here this is the bronchioles. Okay. So you can see here the bronchioles also have a little bit of cartilage there, and actually just a little side note, the very end bits of the bronchioles have no cartilage. So this is the kind of larger bronchioles, and then we have this bit down there, that's the very end of the bronchioles. We actually call those a respiratory bronchiole, which is at the very end. And the difference there is that is where gas exchange occurs. So gas exchange, where does it actually occur? Well, let's highlight, okay? So it happens down here at the respiratory bronchioles once we've got past the cartilagey bit, and it happens, of course, all over here, which is the alveoli, okay? So that, oops, got a bit funny my drawing there. That is where gas exchange occurs. Couple more things to label here. What we got? Okay, so this is the, is it called the pleural space? I forget. It's the space between the two pleural membranes. So there's an inner and outer membrane surrounding the lungs. There's one membrane sort of just over the surface of the lungs, another membrane just inside the surface of the ribs. So I think this is called the pleural space. You might have to fact check that for me. I don't believe it's not on your syllabus, you don't need to know it, but it's the space between those two membranes. So we have pleural membranes here. And actually, if one of these membranes gets punctured, this is what happens when you get a punctured lung. Because what happens if you get a punctured lung is, I won't draw it on the diagram for confusion, but just imagine if you put a hole there, then when you try and inflate your lungs, actually what happens is you suck air into the pleural space. And you end up inflating the pleural space with air as opposed to the lungs, which can mean that you don't do gas exchange and then you kind of suffocate. Okay, I think that is it. Let's now look at some of those structures in a bit more detail and talk about what happens. Okay, so first of all, bronchus. Okay, that's actually up here. This diagram here is supposed to be the bronchus. And this image here shows the tissues in the bronchus. Now this diagram is in your textbook and it's on page 175. but I'll label. So if you want a clearer view of it, go to your textbook and look at page 175. But I will label those tissues here. Okay, so this one here, my diagram's a bit blurry. Maybe you can see better in your textbook. There are goblet cells there. And they secrete mucus, okay? And next to the goblet cells, working in tandem with them, are ciliated epithelial cells. Which, remember, have the cilia can sort of move. So what that happens is what those two things do together is when you breathe in and you inhale maybe dust particles or maybe your bacteria, for example, those particles can get stuck in the mucus. And then the cilia in your trachea, they kind of beat in synchrony. So they kind of move like this. They kind of almost like a conveyor belt. They conveyor belt all those particles up to the top of your trachea, where you then swallow them. Okay? You swallow those particles and they go into your stomach acid and they kind of get neutralized. So it's a defense mechanism. What else have we got there? Well, we've also got muscle. Change color. So these two things kind of almost a defense, kind of linked to the defense systems of the body, which we're going to look at later. So we've also got smooth muscle. And that can constrict the airway. And then underneath we've got connective tissue. Small blood vessels. This is there. You can just about see the red in the middle there. Small blood vessel. And down here we've got the cartilage itself. Now the cartilage, it looks quite lightly staining. That's because a lot of it is not, the sort of cells secrete the proteins that make the cartilage. So all this kind of white stuff would just be the proteiny kind of scaffold, the extracellular scaffold in which the cells that manufacture the cartilage sit, okay? Okay, so that is the bronchus. The bronchus is there, and the diagram of what it would look like under the microscope is there. Okay, if we go a little bit further down, so remember we've got bronchus, then we go into the bronchioles. What would the bronchiole look like? Well, the bronchiole looks like this. This is the diagram of the bronchiole. Again, this diagram is on your textbook page, but let's label. Let's do green. Okay, so we around the bronchiole here, these spaces, these are the alveoli. So this is the alveolus wall, which isn't really part of the bronchiole, it's just the surrounding stuff. So here's the bronchiole going through the middle of those alveoli, and we've got a smooth muscle. Then we've got some loose tissue, some loose connective tissue. And then we've got these here are ciliated epithelial cells. Now, just a little side note on the smooth muscle. So this smooth muscle tissue in the bronchioles can be responsible for asthma attacks. So in asthma attacks, the smooth muscle can constrict. For example, if you inhale smoke, the smooth muscle will constrict the airways to try and stop the smoke getting down into the alveoli where it can do most damage. So smoking causes the smooth muscles to constrict, but asthma can basically happens when the smooth muscle of the the smooth muscle of the bronchioles constricts inappropriately. So it narrows down so that the bronchioles become really narrow, and it becomes then difficult to get air to the gas exchange surfaces, which are the alveoli. So inhalers act to relax the smooth muscle, okay? Asthma equals constriction at the wrong time. Okay, so and then when you have inhalers can target that and the smooth muscle to relax it. Okay, so that is the smooth muscle. Going further, so remember we go trachea to the bronchus, to the bronchioles, down into the respiratory bronchioles, the final stages, and then to the alveoli. So let's look at the alveoli. So here, the alveoli kind of look like a bundle of grapes. So this is these two here would be alveoli. Two of them, one there and one there. Each sort of air sac is an alveoli or / air sac. And then this is the network of capillaries around it, okay? And this network of capillaries, if we zoom in, it kind of looks a bit like this. Alveolus, or the air sac in cross section. And this is really where the gas exchange occurs. So air moves into and out of the alveolus during inspiration and expiration. So this is air going in and going out of the alveolus. Now, when an alveolus, they do change size slightly. So a couple facts about the alveolus. There's around about 700 million in your lungs. Ish. That's an average, obviously. 700 million alveoli in your lungs. That's a lot of airspace. And together, those alveoli membranes, that total area for airspace, if you lay it out flat, it's going to equal about 70 meters squared of surface area together. That's a lot of surface area for gas exchange. Now, typically, the diameter of the alveolus ranges from about 0.2 millimeters to 0.5 millimeters. So that's 0.2 when deflated and 0.5 millimeters when inflated. So 0.2 millimeters, remember, is also 200 microns. 0.5 is 500 microns. So here we have these cells here are called squamous epithelium, squamous epithelial cells. And basically this squamous epithelium means super flattened, okay? Really, really flattened, really, really thin. So very flattened or thin. And they're kind of really squished together in reality. This layer here and this layer here are squished so close that the membrane is incredibly, incredibly thin. I just had to Wikipedia check this, and I actually saw that it said it is between 0.2 and 0.6 micrometers, which is, you know, really, really small. So, you know, around about the order of 1,000th of a millimeter total diffusion distance across those two membranes, which is so small. You don't need to know that, but just very, very thin. Okay, now let's look at what that would look like in a microscope image. So here's a microscope image showing through the alveolar tissue. We've got multiple alveoli here. So let's kind of look at this. So each one of these, this like around here would be sort of one big old air sac. So apparently there is a blood cell. I can't see it myself, but apparently there's a blood cell there. Maybe it's just in there, and you can't really see the capillary wall there, so it's difficult to tell where the capillary is, but there's a blood cell and a very small capillary there. Here we've got an airspace. Here we've got the wall of the alveolus, which is the squamous epithelial cells. So I'm just going to label that like that. Okay, and then in there we've got a tiny little blood vessel. You can just about see in there, kind of sort of circle it like that. Got a blood vessel capillary. And I believe, although I'm not sure, that this here would be a larger, it looks to me to be a larger blood vessel. Now, I want to take you briefly to an image which you haven't got in your mind map, just for sort of this extra detail image I've got here. So this kind of shows a bit more detail of how the respiratory bronchiole, the bronchiole ends into the alveoli. So here we've got the bronchiole, if you just follow where I'm pointing. Here's the bronchiole. It goes into these end bundles of grapes, the alveoli. You've got the individual air sacs like that. This shows the cutaway, showing how they're all interconnected. And we can see the blood vessels kind of wrapping around them. We've also got two key important things here, which I wanted to highlight one more time. We've got the smooth muscle. So these kind of red bands represent the smooth muscle that can constrict to narrow the airway. The space in the airway is called the lumen. I'll label that on the Mindmap in a second. And also we've got these green fibers here are elastic fibers. Okay, so those green elastic fibers help reinflate the airways and the alveoli if the smooth muscle has constricted. Remember, muscle can only work in one way. You can only contract. You can't expand muscle. So the muscle constricts, and the elastic fibers allow the recoil of that. Okay, so that's in extra detail. Let's just pop back to the mind map to label that. Okay, so alveoli airspace. Okay, so connective tissue I wanted to label here has elastic tissue. So that's all over the place. Okay, there's elastic tissue in all the connective tissue. Plus here, elastic fibers, even over here as well. And we can say, by constriction, I wanted to also say narrows the lumen, which is the internal space. Okay. All right, so let me just zoom out so we can see that whole process. Now, we haven't really talked about the inflation of the lungs, how it actually works. Two main mechanical things that cause the inflation of the lungs. Hopefully you remember this from GCSE or even from Key Stage 3. We won't spend too much time on it. But this diaphragm here, oh, I wanted that to be in pink. Yeah, so this diaphragm moves down, okay? So diaphragm, so when it's relaxed, it's up, and when the muscles contract in the diaphragm, it pulls down. So contracts, pulls down, and when it pulls down, it increases the volume within the lung cavities, which decreases the pressure. So increase the volume, decrease the pressure, and then air flows in. So let's just write there, decrease pressure, air in. Okay, so that's inspiration. Expiration, the opposite happens. The diaphragm relaxes, it kind of springs back up, again with sort of elastic properties, and the pressure gets increased in the lungs and air moves out. Also, the rib cage, staying with this pink color here, we can move, let's do sort of like this. So rib cage up. So that's when you're really inhaling kind of quite aggressively when you're exercising, for example. The rib cage can move up. That's the external intercostal muscles. There's two types actually, internal intercostals and external intercostals. So the external intercostals contract and lift up the rib cage for inspiration. And if you really want to force your breath out, the internal intercostals, which are inside the rib cage, they contract and pull the rib cage down. So I've just labeled this rib cage up, external intercostals, and diaphragm down. Those are the two things that cause inspiration, and it's the opposite for exhalation. Okay, so what I'm going to do now is I'm going to try and find a little video to sort of show you that process, all those sort of processes going on in one time, which I'm just going to drop in here. So watch that video now, and then we're going to move on to talk a little bit more about ventilation.
Your lungs are two spongy organs in your chest. The left lung is divided into two lobes, or sections, and the right lung has 3 lobes. When you breathe in, air enters your nose or mouth and passes into your trachea, or windpipe. At the carina, the trachea divides into 2 bronchi, then branches into smaller bronchioles. The bronchioles end in tiny air sacs, or alveoli. Here, the oxygen in the air you inhale passes into the bloodstream, and carbon dioxide from your body passes out of the bloodstream. The carbon dioxide is expelled from your body when you exhale. Your lungs are encased by pleura, a thin membrane that protects them and helps them slide back and forth as you breathe in and out. Underneath your lungs is the diaphragm, a smooth, thin muscle that helps your lungs expand and contract as you breathe.
Okay, so I hope you found that video helpful to kind of bring all these kind of labeled diagrams together and make it seem a bit more lifelike. So we're going to move on to look at how we can measure how the lung works, and measure the inspiration and the expiration, movement of air in and out of the lungs. We do that by using a device called a spirometer, which is shown here in this diagram. So we're about halfway through the video. So if you'd like to pause now, get a cup of tea, then now would be a good time. If not, we're going to plow on, talk about measuring lung volumes, and then we're going to talk about other organisms. Okay, still with me? Let's do spirometry. Okay, so this diagram is in your book, page 176. And we've already labeled most of it, but let's just focus on one or two things. Okay, so the first thing is actually about this person, okay? So when we are going to measure spirometry with a person, we need to make sure that they are healthy. Not safe to do it if a person has asthma, for example, or particularly bad asthma. Disposable mouthpiece has got to be sterile, because otherwise we could have transmission of infections. This chamber here that's filled with water, not overfilled. Otherwise, basically water could kind of get out and into the tubes here, and you could find yourself inhaling water and choking on water. The oxygen's got to be sort of fresh air and fresh oxygen. Otherwise, a person could suffer from hypoxia, which is when they kind of run out of oxygen and their lips turn blue, and that's not good. And of course, the soda lime should also be fresh. And the function of the soda lime is it absorbs CO2. Now, the reason we're getting this the reason for absorbing CO2 is because CO2 is toxic, and if this person's breathing into and out of this device here and CO2 is building up, then that would actually cause them to change their breathing, and it'd also be toxic if it builds up to very high levels. And actually, when we are absorbing CO2, we can use that to kind of measure oxygen uptake as well by this person. We're going to look at this in this diagram over here. Okay, so person breathes into and out of this device, and as they breathe in, this is a little bit confusing here, so I'm going to try and explain it carefully. As the person breathes in, air comes into this. Where's the air coming from? It's coming from this tank. Okay, so as they breathe in, that means that this tank is going to move down like that. It's going to hinge downwards like this as they breathe in, and as they breathe out, back into the tank, it's going to inflate. So this pen here goes up, it goes down when they inhale, up when they exhale, down when they inhale, up when they exhale. So this produces a trace on a piece of this. In an old school spirometer, they had a piece of paper that was slowly spinning next to that pen, and the pen basically, I'm going to try and demonstrate this with a piece of paper here. So as this trace sort of slowly moves, the pen is just kind of drawing on the piece of paper that's slowly moving past it, okay? Obviously now we use digital technology. This is very archaic, which means old-fashioned. And then they sort of flip the paper upside down so that the diagram that's produced looks like this. Okay, so this is called a spirometer trace. Okay, now do we need to label anything up here? I suppose here, up equals exhale, down equals inhale. We could label that. But then they flip the trace upside down so that over here, this represents inhaling and exhaling. Volume of air in chamber. I'm not convinced that that's correct. Yeah, I'm not convinced this is correctly labelled on this textbook here. I think this should not be volume of air in chamber. I think this should be volume of air in lungs, which can be worked out from the chamber. Okay, the reason being the residual volume down here is not a measure of the air that's left in the chamber, but a measure of the air that's left in the lungs. Okay, so I think that's incorrectly labelled on their pot. Bad work. Textbook people. Okay, right, let's label the rest of these things, okay? So as we breathe in and out, normally, just practice breathing out in and out normally, just like this. Now, you're probably doing it, hopefully, without even thinking about it too much, and you're not putting a lot of effort into it because you're probably sitting still. And under normal conditions, the ventilation that we get like that is called the tidal volume. So I'm going to pinch in a bit on this diagram here. And this is called the tidal volume. And that's your kind of everyday breathing, okay, tidal volume. Now, going from your tidal volume, you can breathe out. Breathe out as far as you can. Now, even when you've breathed out as far as you can, this is forced expiration, okay? So this here is, let's label that, okay? So forced expiration all the way. Even when you breathe out all the way, there's still going to be some air in your lungs. So that residual volume is the air that still is in your lungs, even when you think you've breathed out as much as you can. So I'm going to go from tidal volume to forced expiration all the way down, and then I'm going to go back to normal. So there, I've now inhaled as much as possible, okay? And then I could exhale as much as possible. Inhale as much as possible. Back to my tidal volume. So what I showed you there were some other key things that we need to label. This here being the vital capacity. So vital capacity is the total volume in your lungs between your maximum exhale and your maximum inhale. So we could label that here. Max inhale. And down here is your max exhale. Okay, and then the tidal volume is your kind of normal inhale and exhale between those two volumes. This here, this whole thing here is your total lung capacity. But it's difficult to measure your total lung capacity because there is that residual volume that is always in your lungs, okay? Right, let's put a few numbers on these. Let's go to a different color. Okay. So vital capacity. What is this? So this is typically between 2.5 to 5 dm cubed, or liters, okay, dm cubed. Residual volume is around about 1.5. And of course these figures vary. And tidal volume is typically at rest 0.5 dm cubed. Yeah, I mean you can actually get these from the scale here, but at rest typically about 0.5 dm cubed. And it depends what you're doing. Your tidal volume when you're walking versus when you're sitting is going to be different, for example. And vital capacity is going to be affected by a lot of things. So let's talk about the things that affect vital capacity. Fitness / exercise is of course going to affect that. Age, size, and gender. Okay, all those things are going to affect it. Yeah. Oh, and also their height as well, because that's kind of to do with size, but height as well. Okay, so those things are going to affect those lung volumes. Okay, so hopefully that has clarified that. More information on page 176, 177 if you want to go over a bit more detail. And the other thing we can do using a spirometer trace over a longer period of time is measure the rate of oxygen uptake. So this is the same thing, okay, but it's longer time, and you're getting your person just to kind of do their normal tidal volume. Okay, so you're not asking the person to max inhale, max exhale. You're just asking them to breathe normally over a period of, in this case, 60 seconds. Just TV, okay? Or I suppose if they were doing some exercise, you could measure them doing exercise over a longer time, but you're not getting them to do different types of breathing at different times. You just might want to measure their rate of oxygen uptake doing different activities. So what's going on here? Well, I'm going to zoom out a little bit, okay? Okay, so in this scenario, we're talking about the chamber would be filled with oxygen to do this sort of kind of experiment. Okay, so chamber filled with 100% O2 at the beginning. Okay, so when the person is breathing in, they're taking in oxygen. Oxygen is being exchanged in the alveoli, going into their blood. The cells are using oxygen in respiration, and they're replacing it with carbon dioxide. So the carbon dioxide travels back to the lungs, goes out into the air. But then, if you look at this diagram here, as the exhaled air, it goes through this soda line and it absorbs the CO2. So we don't get any extra gases being added back to the chamber on exhalation. So we're only taking out, we're not adding back. So that causes the volume of oxygen, in the chamber to decrease. So now we can measure that rate of decrease. So we pick a point. It could be at the top or at the bottom. In this diagram, it's the point that is being chosen is A. Okay. Now we have to pick a point on the trace that is clearly recognizable. So we would typically pick either the very bottom or the very top. So now we pick another point on the same point on the trace down here, B. And then we have to use the axes here to work out what those values are. Now, let's just say, for ease of my calculation purposes, that A is at 4 dm cubed at 30 seconds, if we go down here. And let's just say that B is at 3 dm cubed, what nice round numbers, at 60 seconds. So what can we work out? Well, we know that the volume of air has reduced from 4 to 3 litres over a time period of 30 seconds. So we're looking at the change. So change, equals 4 minus 3 dm cubed equals 1 dm cubed. And that's the change in volume. Time equals 30 seconds. So we can have a rate of uptake, rate of oxygen use is equal to, well, if you want, you could do it either as 1 divided by 0.5, and that would give you 2 dm cubed per minute, because I use 0.5, 30 seconds is half a minute. Or I could do it 1 divided by 30, and that would give you a value of dm cubed per second. I haven't got my calculator with me, but that's going to be around about 0.33, isn't it? I think. So check the calculator, but then you could get a value of dm cubed per second as well. Okay. All right. So that is everything about spirometry. Have a check over page 176, 177. Check I haven't missed anything there, as I'm sort of running out of time for this video, but I want to spend some time on fish and insects because I find that really quite interesting. Okay, now let's talk about the gas exchange system in fish. And I think one thing that's really interesting about fish is they have basically a better system than us. Okay, their system is more effective at extracting oxygen from seawater than ours is at extracting oxygen from the air. So why do they have to be so good? Well, the reason is there's less oxygen in seawater than there is in the air. In air is a mixture of gases, and it's 20% more or less oxygen. In the sea, we're talking about oxygen that is dissolved in the seawater, and there's a lot less available. It's in the range of a few milligrams of dissolved oxygen per litre. So fish have to be able to extract as much as possible of that oxygen from the seawater and get it into their blood. Now, they do something using a system called a countercurrent flow. I'm going to be looking at that in a bit more detail later, but first let's label this lovely diagram of a fish, which you can find on page 178. Okay, so, right, let's go. Water. Water entering the mouth, so that's water flow. And then here we've got, this is the mouth, not labeled on your diagram, and let's label it here. So that is the mouth. And sometimes that is called the buccal cavity. And then this here is called the operculum, which is the outer covering, which covers the gills. It's kind of like the cheek of the fish, I suppose. And that covers the gills. And when water flows through a fish's gills, it flows through the mouth, through the gills, and then there's a sort of slit here, so it comes out there. So let's also sort of label that. So water goes in this way, and then it comes out to the side. Okay, so there is a one-way flow of water in the mouth and then kind of out slits kind of roughly here on the fish. That one-way flow is going to be really important. Inside the fish's mouth, there is a system that holds the gills in a very specific orientation so that water flows over it in a one-way flow. So that system begins with something called the gill arch, which is this here, GA, gill arch. And in that gill arch, we've got blood vessels. You can see one is carrying oxygenated blood, and the one is carrying deoxygenated blood. So the deoxygenated blood goes to the gills. The gills pick up oxygen from the water, and then oxygenated blood goes back to the fish. Now, the gills has got a system of lamellae, which just basically mean, what does lamellae even mean? Kind of layers or plates or, I don't really know what the definition for lamellae is. Google it, I'm not really sure, but we call them lamellae. We have primary lamellae and secondary lamellae. So these are the gill filaments. Gill filaments, which are sometimes called the primary lamellae. And then if we look at those filaments individually, so like you can see the zoom in scale here, along that filament there individually, there are many of these secondary gill plates, and they're sometimes also called secondary lamellae. So these are gill lamellae, or sometimes called, I'll just do the symbol there, secondary lamellae. Okay, so water flows in a one-way system across the gill plates, so that if we look at the blood flow, this is deox, I'll just deox for deoxygenated, deox blood comes in, and then ox blood comes out. Okay, so the blood is flowing always this way. We can see that here. The blood here starts off, if you look at these numbers, it's a bit blurry here, but can you see it? The blood starts off when it enters the gills as having very little dissolved oxygen, only 5% dissolved oxygen. And as it moves across the gills, it absorbs oxygen from the water that's flowing past it, such that it leaves carrying sort of 90% oxygen on this side over here. Okay, so if you couldn't see that, just maybe write those numbers in. Let's just write them in underneath, actually. So let's sort of go 5 to to 30. These aren't exact numbers. It's just showing that the oxygen is being absorbed. And let's do that by changing the kind of arrow like that. And this is the blood. Okay, so the blood goes from deoxygenated to oxygenated. It's picking it up. So this is the blood flow. This diagram here, this shows blood flow. And the other one is the water flow. Let's make those stand out a little. So I'll just highlight water flow in blue, and I'll highlight blood flow in pink. Okay, so water flow and blood flow, opposite directions. That is very, very important. Right, I'm going to show you a little animation now. I'll leave this diagram there, and I'll show you a little animation so you can relate that to why it's so important, countercurrent flow. Okay, so here's a diagram showing those primary and secondary lamellae in a bit more detail. And it shows quite nicely how the gill filaments have the secondary lamellae or the gill plates on them. So you can see that here. So here, once again, we see that the water flows in one direction whilst the blood is flowing the other direction. Now, I don't actually like this here because the blood should be going this way. This arrow is not really good. Both arrows should be going from right to left across. Now, let's see why that's so important. So, first of all, let's look at, this is the diagram again. First of all, let's look at what would happen if the water and blood were flowing in the same way, okay? This is not what happens, but it's just to see what would it be like if it was set up this way. So in this diagram, water and blood are flowing the same direction. Okay, so if we kind of run this animation, and we start on with water carrying sort of 9 milligrams per litre of oxygen. Okay, that's actually around about the accurate value. And the blood starts off carrying about 1 milligram per litre of oxygen. Right, as they flow across the gill filament together, the gill plate together, sorry, they're going to equilibrate, which means that by the end, they're going to have roughly the same amount of oxygen. The water has gone from 9 to 5, the blood has gone from 1 to 5. So the blood reaches a saturation of 5 milligrams per litre, or about 50% saturated. Okay, so how would that be different if we ran the blood and oxygen in the other way? This is just another a view of a graph of that. So you don't need to draw this graph. You can if you want to, I suppose, but this would be if they were flowing the same way together, we could represent this in a graph like this. So this being distance along the gill, and this being the saturation of blood with oxygen, which starts at 0 and climbs to 50%, and the water starts at 100 and falls to 50% where they reach an equilibrium. Now let's look at what happens if we do counter current flow. So this is what actually happens. Now here you've got to sort of look, the water is going from here to here, and the blood is going from here to here. Okay, so let's put some numbers in. Let's start the animation. Well, it's difficult to look at it piece by piece because the blood is flowing past each other simultaneously. But let's look at the blood first. Okay, so let's start over here with 1. Blood that enters the gill, carrying very little oxygen, 1. It is in contact with water that's already flown through most of the gill, but that water's still got a bit of oxygen to extract. So there's still a concentration gradient here. So the 1 or the 3 here can still get some oxygen from this 4 and 2. There's still a gradient going down. As the blood flows more towards the middle of the gill, it's meeting water that is fresher, that has higher oxygen concentration. So it can still absorb more oxygen. And as the blood flows right to the beginning of the gill plate, it's meeting the freshest water that has just entered the gill and is carrying the maximum amount of oxygen. So it can still absorb oxygen. So notice the blood here can reach 9, whereas before it could only reach 5. On a graph, we could represent that like this. So the blood is flowing from bottom right to top left, and the water is flowing from top left to bottom right. So this graph is on your mind map, so let's now label that now. Okay, so here's that diagram. We've got the blood flowing this way, the water flowing this way. I suppose there's not much that we need to do in terms of the labeling, but I think we should say that blood exits gill having, let's say, extracted most of the available oxygen, most of O2 available. And here's a nice phrase that we can use. Countercurrent flow. So that beeping is my washing machine, it's finished. Maintains a concentration gradient all the way along the gill plate or the gill lamellae. Okay, so all the way along this, there is a not constant, but there is a maintained gradient, a difference between the water and the oxygen, which allows more oxygen to be extracted. Okay, so that is the fish. Fantastic system there from the fish, one of my favorites. And now let's go on and look at the insect, which is an entirely different system. We haven't really talked about ventilation, and I'll keep the same colour that we used before. So what about ventilation? Do fish breathe? I mean, when I'm talking about breathing, humans, we breathe. That's the physical movement of air in and out of our lungs. Do fish do that? Well, not really. They can. It depends, actually. There's two mechanisms of ventilation. Number one, movement, okay? When fish are swimming forwards, they have their mouth open, okay? Water flows through their mouth and out their gill slits, and that just moves water across the gills. Some fish, mainly sharks, can only do this, okay? So, for example, great white shark, they are what's called an obligate, this is not on the syllabus, but let's just say it anyway because it's cool, obligate ram ventilator, okay? So that means that they have to keep swimming, otherwise they can't get any water across their gills. They have to keep swimming, and the water is rammed through their open mouth and past their gills. They're an obligate ram ventilator. But some fish can kind of do that. Okay, so they open their buccal cavity, that's their mouth, and then they close it. And when they close it, they open their gill flaps. When they open, water goes in their mouth, and when they close, water comes out their gills. So they can also do that. So open plus close the mouth so that water moves in the buccal cavity and then out the gill slit. Okay, so that's how fish ventilate. All right, final thing, going into insects. All right, insects. Insects have a really, really different system. And one of the reasons that it's so different is insects' circulatory systems are also very different. And what I mean by that is that they have an open circulatory system. We're going to look at this in more detail in the next Mindmap. But they don't have really blood vessels. So they don't have blood either. They have something called hemolymph, which is a blood-like fluid, and it just circulates in their body cavity. So instead of having an organ which supplies the blood or the hemolymph with oxygen, they sort of have a distributed system. So they don't have blood vessels that supply blood to all parts of the body. What they do have is they have the tracheal system that supplies oxygen via tiny little kind of tubes of air to all parts of the body. It's kind of a reverse of our system. So let's look at that, but first let's label this insect that we got here. All right, okay, so insects, they have often a thorax, that's the main kind of, this is the head obviously, head here. Thorax is this section here, and they have an abdomen here, which is their kind of rear section. Now, if we zoom in here, we can see this little red network here consists of spiracles and trachea. So spiracle, first key word, that's the opening, and the trachea is the tube. So here, zoomed in at this part right here again, we can see this enlarged surface. So this is the surface of insect, exoskeleton. Let's even say exoskeleton. Here we have the spiracle, which is an external opening or pore. You can add the definitions here actually from the book. External opening or pore that allows air into and out of the trachea. Here we have the trachea. Now remember I said that with the human trachea we had cartilage to support it and to stop it collapsing. Similar sort of thing going on with the insect. The insect trachea is supported, chitin gives support. And I believe the exoskeleton is also made of chitin. That's a material, a biological molecule that we don't really have. Chitin is a mammal that gives the insect exoskeleton support and also the trachea. Then we have kind of smaller tubes that branch off this trachea here, and we call those tracheoles. Change color, tracheole. And then down at the final ends of these narrow tracheoles, here we actually have the site of gas exchange. Okay, so we deliver oxygen via the spiracle. So air, let's just trace out the flow of air here. Goes through there, down here, down through the trachea systems, down the tracheoles, all the way to those final sites of respiration. So basically, insect, if you do an insect dissection, you find these tiny little white tubes everywhere. They tend to be white, the ones I've done anyway. And those white tubes are the tracheal network. So they supply oxygen all the way to the cells by this tiny little network of tubes that kind of go branching out through the insect in a similar way to blood vessels do in a human, except remember, there's no liquid in these. It is air in these tubes. Now, some insects can actually have a slightly modified version of the system because they're very active and they need to get a lot of oxygen to power their flight muscles, specifically if it's a flying insect. So in this So using this system, they can change their kind of rate of oxygen uptake by modifying the surface area of their exchange system. So let's label this. Okay, so this shows here. Okay, so we can modify the kind of the structure of the tracheal system by moving tracheal fluid. So this would be under low demand. Okay, so if the insect maybe is not flying, then its tracheal fluid kind of sort of leaks into the tracheole, so that the total surface area, if I sort of highlight in blue here for gas exchange, is this much. Okay, so that would mean that would have some benefits because it would mean that the insect would probably be losing less water out of the spiracles by having a slightly reduced surface area. But under high demand, the insect can withdraw tracheal fluid. So the fluid kind of comes out into the tissue. So now if I highlight that surface area, we have more surface area. You can see how I'm highlighting more blue. So greater surface area, larger surface area for gas exchange, which means that we can absorb more oxygen. It might have a consequence that the insects might desiccate, dry out a bit more quickly. Okay. Furthermore, for ventilation, I'm going to keep my same pink for ventilation. I don't know why I picked that, but so can insects ventilate? Some, okay, some can. Extremely sort of active insects can. They can flex their abdomen to kind of increase ventilation, and in some insects, the movement of the flight muscles actually increases the ventilation into and out of the trachea, which is nice because when they're flying they need more oxygen, so the flight muscles kind of help with the ventilation. And locusts have specialized breathing movements of their tracheal system as well, which involves kind of opening certain valves, moving air down the system, and then opening other valves to let it out. So there's a bit of a kind of a one-way flow of air through the system. So just to summarize that, ventilation, we can expand, contract body segments, we can have flight muscles, help ventilation, and locusts can open and close specific combinations of spiracles. Okay, I think that's everything. I'm just going to zoom out so we can sort of reflect on what we've covered, which is a lot. Sorry, this video has taken just a little bit over an hour, but I think we've got everything we need there. That's it. Okay, so very quick review. We've talked about the need for exchange systems, talked about surface area, size, shape and level of activity. We said the features of a good exchange system are always large surface area, short diffusion distance, and good blood supply. Then we looked at the lungs in detail, looking at the tissues, the types of images that you might have to analyze. When we looked at spirometers and the graphs that we can get from them to look at aspects of ventilation and the rate of oxygen uptake. Finally, we've looked at fish and insect gas exchange systems. Okay, I hope that's been helpful to you. The next thing I'm going to set you some multiple choice questions on this, followed up by a look at the circulatory system on our next mind map. Thanks very much. Bye-bye.